This,
Yeah, I think that's a good angle. I think people like to have abbreviations.
yeah,
The lambs, yeah.
yeah,
Like TAMS.
Lambs, lambs.
Yeah. Yeah. So, all right. Well, it's 8 o'clock. I guess we should begin. Jennifer, do you have any announcements or anything to make?
No, I think we can turn it over to you, Judy, for the acknowledgements and introduction.
All right, everybody, welcome to today's Myeloid Network meeting. We're thrilled to have a fantastic guest speaker, Susan Kaech. I want to again acknowledge that we have support from the University of California Moores Cancer Center, AstraZeneca, and that we
Prisma Therapeutics.
yes, thank you. And we're developing new websites, so we hope to get that up and running shortly, where we'll have an interactive site where you can post comments and have discussions online about macrophages and topics related to myeloid cells, macrophages, and related cells. So today I'm pleased to introduce Susan Kaech, who's a professor at the Salk Institute in San Diego, California. She's the director of the NOMIS Center for Immunobiology and Microbial Pathogenesis. She's a distinguished immunologist who's been working in cancer immunotherapy, in particular metabolism. She is a professor at Salk, and where she leads cutting edge research exploring how memory T cells develop during infections and vaccinations, but also, as you'll hear today, she's doing groundbreaking work on macrophages as well. She earned her Ph.D. in developmental biology at Stanford University and did a postdoctoral training with Rafi Ahmed at Emory, and then joined Yale University, where she was a Waldemar von Sedgwick's professor. She came to San Diego a few years ago at the Salk Institute, where she has performed groundbreaking research on metabolism in immunity, and recently was inducted in the American National Academy of Sciences. She's also received other notable awards, such as the PKS Award, and we're thrilled to have her today to talk to us about the work on metabolic co-option of resident macrophages in lung cancer. So as you can see, her title, Co-opting Tissue-Resident Macrophages in Cancer Through Metabolic Reprogramming. We're thrilled to have you today, and look forward to hearing your talk. Thank you, Sue.
Thank you so much, Judy. And I just want to also thank you personally, because you've been such a welcoming colleague coming to San Diego and creating this network. And really, for me, a lot of you probably don't know who I am because I'm not that well known for my work in macrophages. I started my career studying T cells and have started to turn my attention towards macrophages for many reasons. But we have quite a heavy investment in the lab now on macrophage-related projects. And I think it's quite interesting thinking about some of the parallels between T cells and macrophages as we think about tissue adaptation and tissue residence. And that's really has been one of the connections that has helped me to transition to focus more on macrophages in different tissues and different contexts. First, I just want to start off by thanking the people who did the work I'm going to talk about today. I'm going to talk about some work that was published this past year, but I figured unread and unpublished might be pretty synonymous, as we have so many papers that come out each week that's hard to keep up with. But I thought I really love this story and this project. I think it helps provide some fascinating paradigms and concepts to think about as we think about the role of macrophages, especially in cancer. And this work was carried out by a fabulous grad student, Allie Coleman, who's up here at the top. And this started as a collaboration with Katie Politi at Yale. And we've also had a lot of work from Zoe Zhu. I'm not going to talk about her work today, but she also has had a central macrophage project in the lab, and her paper will be coming out very soon in cancer research, actually. And so, yeah, so this was just a lot of great work from these strong collaborations with Ali, Katie, and Zoe. And so I don't need for this audience to tell us the importance of and what tissue-resident macrophages are. We know that they play specialized roles in the tissues that they inhabit and I think, the important thing I usually try to tell T-cell audiences when we're talking about tissue-resident macrophages is that these cells were actually present at the time that the tissues were being patterned and as the tissues were developing throughout development. And I think that's an important just note to take because that means that they really were listening to tissue specific cues and understanding the specialized functions of those tissues, those different organs to help carry out those functions of those organs. And to date, my lab's been focusing on a few different tissue-resident macrophage populations. We've been looking in the brain at microglia quite a bit. We published a paper a year ago or so on the role of microglia in helping to present tumor antigens and serving as APCs in glioblastoma. And that was helping to provide a therapeutic protective response for CD4 T cells in immunotherapy for that the glioblastoma was responsive to. So that's one area, and we're still looking a lot more at microglia now in the context of aging and inflammation and Alzheimer's disease as well. And what I'm talking about today, though, is our work looking at the role of alveolar macrophages in lung and in lung cancer. And the concept that Ali and I really started on was this one that I kind of already introduced, which is how tissue-resident macrophages, the roles that they serve in tissues for their client epithelium. And I'm using terminology that I think has become quite common in the field now, first introduced to my knowledge by Ruslan Medzitov, thinking about the supportive functions, the homeostatic functions that these tissue-resident macrophages have within the tissue, but that they really have evolved this homeostatic relationship with their client epithelium, because that's the cell type that they have to care the most about and try to serve and support. And that we normally think about these functions that these tissue-resident macrophages have is involved in maintaining tissue homeostasis and repair. Sorry, that's my dog getting out of the room. But we wondered if these tissue-resident macrophage functions may be potentially these homeostatic relationships that develop. First, we have to understand what those really are mechanistically. What are the molecules and the signals that are being transferred and communicated between the client epithelium and the tissue-resident macrophage? But once we start to understand what those signals are and what those processes are in greater depth, how are these processes then perhaps being co-opted in types of diseases, and in particular in cancer? And could a tissue-resident macrophage essentially somewhat be hyper-controlled or hijacked in some ways by the tumor environment or by the client epithelium that's now become transformed and malignant to alter the tissue-resident macrophage functions in a way and through these what were normally homeostatic pathways and processes, but now can be turned into pathways that would be converted to support tumor progression. And so that was kind of the general concept and paradigm that Ali and I started off with thinking about in the role of lung cancer. And so just to remind you all, a lot of the fundamental roles that alveolar macrophages play in the lung alveoli, we know that they play very important roles in maintaining tolerance and a reduction of inflammation. And that's because the lungs are constantly exposed to environmental debris and pollutants, and these need to be maintained under control, but not and cleaned up, but not in a way that would lead to inflammation. However, on the flip side, if the lung does become infected, it also, these cells have antimicrobial functions where they can help to secrete inflammatory cytokines. They can elicit T cell effector functions and prime T cells, and those are other important roles where they can now toggle towards a more inflammatory state. But this kind of central role, one of the central roles that they play that we know is very self-evident, is in the role and maintenance of surfactant. So this lipoprotein, this very lipid-rich lipoprotein that helps line the air-liquid interface that allows us to breathe, and macrophage, and this surfactant is secreted by the epithelium. So looking at the type 2 epithelial cells here, the type 2 pneumocytes, these are the cells that secrete the surfactant. The alveolus is surrounded by type 1 epithelial cells, and then this very specialized type 2 epithelial cell that secretes the surfactant. And this plays a very important role for maintaining lung health and for, as we were saying, to allow the air-liquid interface to be preserved and for our lungs to expand and to enable breathing. And the alveolar macrophages play important roles in recycling that surfactant, and as well as the type 2 pneumocytes play a very big role in recycling the surfactant, but helping to maintain that surfactant is under high good quality, reducing oxidation levels of the surfactant and helping to maintain and preserve the quality of that. But when tumors start to arise, and this was really kind of the idea that we started to think about with Ali and Katie, was like when the tumor cells start to become transformed and start to proliferate and increase in their mass in the lung tissue, what does the alveolar macrophage do? What does this tissue resident macrophage do? How is it reacting to this? And we've thought about perhaps its normal day-to-day tolerogenic states might enable a more tolerogenic anti-inflammatory environment that could help to maintain more, suppress anti-tumor immune responses, maybe recruit more regulatory T cells. But we also wonder what could be happening with this other metabolic role that alveolar macrophages play in maintaining surfactant and this lipid metabolism, this lipid homeostasis that occurs within the lung. And so that was just kind of a question that we had in the back of our minds. So we thought that we would try to explore this question using a model of lung cancer that Katie Politi developed. She did her postdoc many years ago in Harold Barmas's lab and was one of the first to start to develop these inducible models of oncogenic, of oncogenes. And in this particular case, focusing on EGFR models of lung cancer, which is a fairly common type of lung cancer, of non-small cell lung cancer that arises. And by overexpressing in an inducible manner this oncogenic form of EGFR, it's the L858R mutation. This is found in humans. She was able to, by the expression of this oncogenic EGFR, transform the lung epithelium into a lung adenoma. And so you can see here is the DOX induction. So using doxycycline, you can induce the EGFR. You can see here it's being expressed in the type 2 pneumocytes because this is they make surfactant protein C. And then this is just showing you at kind of an early stage when you first can start to see the macroscopic tumors forming. This is about 4 weeks. You can see by MRI, you can see these lesions forming. And then this is a much later stage. The mice start to succumb around 8 to 10 weeks of induction of the oncogene. And this is an example of a lung lobe that is almost entirely containing tumor transformed cells. And the importance of this model also over the years, as we've started to study the responses of lung cancer patients to immunotherapy, it's become quite clear that patients who have mutations in RAS and some other mutations are much more responsive to immunotherapy. But patients who have EGFR mutations are notably very resistant to immunotherapy. They have lower tumor mutation burdens on average. They usually arise in non-smokers. So unlike the KRAS, the RAS mutations, those are more common to smokers that are more responsive to immunotherapy. But so actually EGFR, people with EGFR mutant lung cancer aren't even offered immunotherapy because we know that they're going to be resistant to it. Now, we don't know what this resistance, what's the basis for this resistance. As we've learned from immunotherapy in general, tumor mutation burden, while being lower, could tilt a tumor to being more resistant to immunotherapy, but that in it alone is not the sole answer. There's something else about the microenvironment that must be different in these types of tumors that have EGFR mutant lung cancer. And so that's a kind of a backdrop that's become much more clinically relevant, as well as we've started to work with these models over the years. And this was the very first result. I have to be quite honest. We started to work with Katie because we were going to start to study T cells in lung cancer. And when we started to profile the immune cells in this tumor model, we saw some interesting changes that were occurring in the major cell types that we could find in the lung. So if you compare it to a healthy lung, this is without any DOX induction, but the same age-matched time points. You can see that we have an abundance of alveolar macrophages. We have an abundance of CD4 T cells and a very small population of CD8 T cells. This is just kind of your resting state lung. But as we started to induce the tumors with doxycycline, we could start to see changes occurring. We saw a steady decline in the CD4 T cells and a modest decline in the CD8 T cells. So the T cells were starting to go down in their density in these lungs over as tumors progressed. However, I should note that the Tregs actually as a proportion were increasing. So even though the total numbers of CD4 T cells were going down, there was a continual increase in the percentage of CD4 T cells that were regulatory T cells. But the big surprise was this increase and a pretty profound increase in the tissue resident macrophages, the alveolar macrophages. We're referring to them as TA for tumor-associated alveolar macrophages. So you'll see this TAAM abbreviation on our graphs for tumor-associated alveolar macrophage, and then we'll refer to healthy alveolar macrophages as HAMs, so HAMs, which is a horrible term, but that's what we came up with. And so, yeah, this was a pretty surprising result. We better take note of this because this increase in tumor-associated alveolar macrophages was quite profound. And we started to look at other properties of them. We noted that they already express PD-L1, but their PD-L1 expression even increased further as the tumors progressed. And TNF production, the ability of these cells to produce TNF, actually went down as the tumors were progressing. So if you remember that model I was just showing you a few slides ago about how these alveolar macrophages can toggle between anti-inflammatory and inflammatory states, this question we asked, would they be more anti-inflammatory as tumors progressed, was actually being suggested and demonstrated by these data here. So there are clearly changes occurring in these alveolar macrophages as they were accumulating in these tumor-burdened lungs. And this is just looking at some RNA expression of other chemokines and cytokines. And so we saw across the board a drop in the expression of many inflammatory effector mediators being produced by these alveolar macrophages as in these tumor burdened lungs. So they were kind of changing their functional states across many different genes. So just to summarize some of the functional data, we saw that as these tumors progress, these alveolar macrophages, these tumor-associated alveolar macrophages started to look more anti-inflammatory. They had low TNF. They actually had high TGF-beta production. They had higher PD-L1 expression, and CD200, which is a well-known receptor on alveolar macrophages for maintaining the tolerogenic properties of these cells, was also increased in expression. So those were some of the phenotypic changes we saw and functional changes that we saw as the tumors progressed in these alveolar macrophages. So thinking then, what would be driving this accumulation of the alveolar macrophages in the lungs? We started to think about the cytokine GM-CSF, and GM-CSF plays a very important role in AM development. We know that it's a necessary factor for alveolar macrophage formation in the lungs. It induces expression of a transcription factor called PPAR-gamma, and PPAR-gamma is also essential for the development of these. And this pathway of GM-CSF induction of PPAR-gamma will lead to intermediate stages of development until you get to a more fully mature alveolar macrophage, which we can define using markers. These cells express actually a high level of CD11C. They lose their expression of CD11B, and they gain the expression of the Siglec F as they become more fully mature. And so we started to think about them, what GM-CSF could be doing, and we looked at GM-CSF concentrations in these lungs as the tumors were progressing, and we actually did see an increase in GM-CSF in these tumor burden lungs. So this seemed like this could be a pretty a likely candidate for driving the progression of this amplification and proliferation of these macrophages. We also looked at IL-4, which would have been another good candidate, and we did not see any evidence of IL-4 levels changing. We couldn't even really detect it, to be honest. And we did try to block IL-4, and we didn't see any effects, but we did see pretty profound effects when we blocked GM-CSF. So we injected anti-GM-CSF antibodies intratracheally, actually. So we were locally trying to block GM-CSF in the lung and in the airways, where the concentration of the antibody would be the highest. And what we found was that there was a very strong depletion of these alveolar macrophages after a couple of weeks of treatment. And these are in the tumor burdened animals. So they did need that GM-CSF to maintain that cell population and likely to drive this increased proliferation since the levels were higher. And what was really kind of interesting, though, is when we blocked with GM-CSF, we also saw a pretty profound reduction in tumor burden in these lungs. So now we're depleting it over the course of we started at week 4 and went on to week 8. And you can see that the magnitude of tumor burden was reduced by about half. And the bar graphs may not look like a lot of difference, but if you look at the physical histology of the tissue, reducing the tumor mass by half throughout the lung it's a pretty profound reduction of the tumor mass. And we also saw a similar effect when we depleted or gave treated the animals with clodronate, which would also lead to a depletion of all phagocytic cells. So we know this is not selective to the alveolar macrophages, but again, we saw a very similar reduction. Basically, the tumors are not progressing as fast. They're growing much slower when we deplete the alveolar macrophages, either with GM-CSF blockade or with the clodronate depletion. So what this told us, which was pretty important, is that this increase in the amplification of these macrophages as the tumors were progressing was important for that tumor growth to occur. So the tumors growth seemed to rely on the presence of these increased alveolar macrophages that had altered their functionality and their states, as we had observed with the changes in their inflammatory functions and the gain of TGF-beta, for instance, and CD200 expression. So the other thing that's kind of interesting to note with GM-CSF is that the sole producer of GM-CSF in the lung are the type 2 pneumocytes, these airway epithelial 2 cells, so these AE2 cells. These are the cells that produce the growth factor to create the alveolar macrophages and to help them develop. And I think this is coming back to this concept of this, what are the actual pathways that the alveolar macrophage, the tissue-resident macrophage, is used to communicate to their client epithelium in the lung. This is clearly one of the most important ones, because the client epithelium produces the GM-CSF molecule that will then control the development, and in this case, the proliferation and amplification of this tissue-resident macrophage population in the lungs. So you have here now the tumor cell as it's expanding, as the client epithelium becomes transformed and now is expanding. The increase in GM-CSF could simply be becoming from the increase in the numbers of type 2 like pneumocytes that are producing GM-CSF. And then that in turn is leading to an increase in the number of the alveolar macrophages that, when disrupted, if we get rid of them, now slow down tumor growth. So we're already seeing this kind of really interesting interdependence between the client epithelium and the tissue-resident macrophage in this process of transformation and progression of lung cancer. So coming back to this role, though, that we know that the alveolar macrophages play in surfactant homeostasis, made us start, and we've been interested, my lab in general has been interested in lipid metabolism in immune cells, both T cells and macrophages. We started to ask, well, what was happening to lipid metabolism in these macrophages? And so we started to profile the bronchiolar lavage fluid of these animals as tumors were progressing in their lungs. And we collected bowel fluid from healthy controls, age-matched controls that didn't receive DOX, or actually they did receive DOX, but they didn't have the oncogenic EGFR, to animals that had tumors progressing. And we could see that we did lipidomics on these. So we're looking at many different classes of lipids here, looking at cholesterols, cholesterol esters, fatty acids, and many phospholipids. And I forgot to mention, but surfactant is a very rich phospholipid lipoprotein. And you can see that there is just a general increase in the overall abundance of these many different classes of lipids in the lung airways of these tumor burdened lungs. So what this told us is that there's a disruption in the normal maintenance of lipid either production or metabolism of these lipids, but there's an overall increase of lipids in these lungs. And if we looked at now how the alveolar macrophages themselves looked at their intrinsic properties and how they were reacting to these lipids, we did some uptake assays. So we looked at how these macrophages were able to import different lipids. And so these are using fluorescent analogs of lipids that are sold like Bodipi analogs, and they have ones that have different lengths of fatty acid chains. And so we looked at long chain fatty acids, we looked at cholesterol, and we looked at phospholipids using these Bodipi analogs, which is a fluorescent-based assay. So you can look at the import of these lipids into these cells, and you can see that the tumor-associated alveolar macrophages had much higher import and uptake of free fatty acids, cholesterol, and phospholipids. But it's important to note that when we looked at the actually at the content of these alveolar macrophages in these lungs, we found that even though they were importing them, we actually found that they contained, when we did lipidomics on the alveolar macrophages themselves, we saw that they actually contained decreased stored triglycerides and stored cholesterol esters. So it suggested that they were importing them, but they weren't really storing them. So they must be metabolizing them. They must be burning them or effluxing them. And that's what I'm going to show you in the next couple slides, that that's indeed the case. So we did some other metabolic assays on these alveolar macrophages that were coming from either healthy control lungs, so that's where this HAM comes in, the healthy alveolar macrophage, or the tumor-associated alveolar macrophages. And when we looked at mitochondrial respiration, we saw that it was really high in these macrophages that were in the tumor-burdened lungs. So not so much by content, actually. The content didn't really increase too much. So this means that these are really revved up mitochondria in these tumor-burdened lungs. And so we worked with Christian Metallo, who's a great, those of you in San Diego probably know Christian very well. He's also a world's expert in lipid metabolism and does a lot of flux analysis. And so we did some tracing of different lipids and of glucose to look at how metabolically active these alveolar macrophages were from these tumor burdened lungs. And we found that there were several metabolic activities that were greatly heightened in these alveolar macrophages, one being the TCA cycle. So in line with having much higher mitochondrial respiration, their TCA cycle was also higher, and we could see increased labeling across many of the TCA metabolites. We also found that these alveolar macrophages, though, were also burning their lipids. This is a fatty acid oxidation measurement that we did, and we can see that there was significantly increased fatty acid oxidation in these alveolar macrophages. Again, this is a process that's occurring in the mitochondria. So the mitochondria have an increase in overall OXPHOS, increase in TCA, and they're also increasing in fatty acid oxidation activity. But what's kind of counterintuitive and surprising is that we also saw that these macrophages in the tumors had an increase in lipid synthesis and fatty acid synthesis. So what this showed us is that they're importing their fatty acids, they're burning their fatty acids, but they're also synthesizing fatty acids. And this kind of made us scratch our heads for a while, but Christian said to me, I think that they're just cleaning the lipids. So we now refer to these cells as lipid launderers because we think that might be a vital function that these cells play. And this is probably, again, a function that they normally play in trying to maintain lipid homeostasis and surfactant homeostasis within the lung alveoli, is this ability to import, burn, process, and even if needed, synthesize new kind of clean types of fatty acids in case these fatty acids are getting oxidized or modified in other ways to help support the general health of the lung airway and the client epithelium. So this was something that we started to think more about, this role that these alveolar macrophages might have in maintaining this lipid metabolic health within the lungs, and that perhaps this was being amplified and really augmented in the context of the lung cancer as these tumors were developing and starting to influence not only the numbers of alveolar macrophages that were present in the lungs, but also their metabolic states. So we referred to this transformation that we saw and these alterations in metabolic activities in these macrophages as metabolic reprogramming, because we saw such a dramatic increase in lipid uptake, OXPHOS, and also fatty acid oxidation. So what could be driving these metabolic changes that are occurring in these alveolar macrophages as the tumors are progressing? And most importantly, are these metabolic activities even important? Is this helping in some way to facilitate tumor progression? And so we started to focus on this transcription factor PPAR-gamma that I introduced a while ago, because it plays such a fundamental role not only in alveolar macrophage development, but also in metabolic homeostasis of cells. PPAR-gamma is a central regulator of lipid metabolism in cells. And there's a lot of already known important roles that it plays, not only in lung alveolar macrophage development. There are people who have deficiencies in PPAR-gamma, and because this will impair their function of their alveolar macrophages and the numbers of alveolar macrophages that these people have in their lungs, they'll develop pulmonary alveolar proteinosis, or PAP, which is associated with severe inflammation and inflammatory macrophages within the lungs. But as I mentioned, we know that PPAR-gamma also induces expression of lipid transporters, can control mitochondrial function, and can play a role in lipid storage. But PPAR-gamma has also been shown to be a role in toggling macrophages between these more inflammatory or anti-inflammatory states. And I hate to use the word M2, but in vitro, it has been shown that PPAR-gamma is required for M2 macrophage activation. And I should say that the Kopf lab had also shown a few years before we started to work on this project that deleting PPAR-gamma in the lung alveolar macrophages would obviously impair their development, but it also led to an increased inflammatory state across many different of the chemokines and cytokines that I showed you earlier. Those were being regulated by PPAR-gamma. So when you lost PPAR-gamma in the alveolar macrophages, you saw an increase in several of these inflammatory gene products, like TNF alpha and some of those chemokines. So that made us think, well, then is PPAR-gamma central to this remodeling or rewiring of the alveolar macrophages that was occurring in these tumor microenvironments? But first, I just want to take a little bit of a step back and talk about some work that's been found in human alveolar macrophages. And we were first really intrigued by a study that came out by Miriam Murad several years ago, where she was one of the first to do single cell RNA-seq and CyTOF on the macrophages and the myeloid cells from patients with lung cancer. And so this is looking at her on the far left, this is looking at her CyTOF panel. And although she didn't call these cells alveolar macrophages, we kind of went back in and dug in and looked at some of the data, and they, by all means, represent alveolar macrophages. And so you can see that there's they could find PPAR-gamma protein in these alveolar macrophages that were increased. And also is interesting, PD-L1 and CD206, which is also another marker that's seen on alveolar macrophages. So we thought that this PD-L1 connection was also kind of interesting to our data. And if you looked at many PPAR-gamma target genes, we reanalyzed her data you could look at the RNA-seq data, and this is what she referred to as the tumor macrophage versus the macrophages from the healthy lungs. And you could see that there was an increase in a lot of different PPAR-gamma target genes in these macrophages from these humans that have lung cancer. And if you look specifically again at PD-L1 and correlated it with the PPAR-gamma high and low macrophages, you could see that there was an increase of PD-L1 on these PPAR-gamma high macrophages. So we thought these were some interesting trends that were consistent with what we were also seeing in the mouse models. So we started to knock out PPAR-gamma in these alveolar macrophages, and we used the CSF1 RCRE to deplete this. We know this will deplete in lots of different, in all macrophages, but we also use, it's important to mention, we also use the CD11 CCRE. Again, that's a unique feature of alveolar macrophages compared to other macrophages in the lung. We know it will also delete dendritic cells. So we looked at the congruence of these two different CRE models, and they were very similar. They had high recapitulation of results between the CD11C and the CSF1 RCRE. So I'm just going to show you the data from the CSF1 RCRE, but we think that the combination of these data, the intersection of these data, tell us that it's predominantly the alveolar macrophages that we're affecting. And I forgot to mention, but when we looked at the lungs of the other macrophages, like the interstitial macrophages, and there are very few of those in the lungs of these animals. So really the alveolar macrophages are the dominant macrophage myeloid cell population. So what we did then was we deleted PPAR-gamma from the macrophages and looked at what happens to tumor progression in this model. And so here's looking at the alveolar macrophage frequency. And again, you can see we have a high number of alveolar macrophages in these tumor burdened lungs, shown here in red. But when we knocked out PPAR-gamma, we reduced this because that's going to impair their development. But what was really nice to see and interesting to see is that when we knocked down PPAR-gamma in these alveolar macrophages, we also saw a reduction in tumor progression. So again, very similar to what we saw when we did the GM-CSF treatment and the clodronate treatments. We're really slowing down the progression of these of tumor growth in these animals that don't have mature alveolar macrophages, as seen with this PPAR-gamma deficiency. We also did another approach altogether, which was to treat the animals with PPAR-gamma antagonists. Now, there's these compounds that have been shown to antagonize PPAR-gamma, and we gave them intratracheally to try to create a more local therapeutic effect. And that also recapitulated many of the phenotypes that we saw in these PPAR-gamma genetic deficient models. So not interfering with PPAR-gamma activity directly through genetic deletion in macrophages or through treatment with these antagonists had a very similar effect on tumor progression and on the changes that we saw within the tumor, the alveolar macrophages. So hold on, I'm having a hard time moving this. Why can't I move this? There we go. Okay. So we wanted to come back now and also see how PPAR-gamma because it plays a role in lipid metabolism in cells. We wanted to see what role it was playing in the alveolar macrophages lipid metabolism, given we saw all these metabolic changes that were occurring. So we looked at cholesterol efflux, and this is also in part because we had learned from some other work looking at lung infections from the Trapnell lab that the cholesterol efflux is increased by GM-CSF. And since we already knew that GM-CSF was playing a role here in regulating alveolar macrophage numbers in these lungs, we wondered if maybe it could be also modulating the metabolic states of these macrophages. And so we looked at cholesterol efflux based on their work and found that indeed these alveolar macrophages in these tumor burdened lungs were also increasing the efflux of cholesterol. So again, we've got like so many state changes that are occurring in these macrophages. We've got an increase in import, an increase in burning of lipids. We have an increase in synthesis of lipids, and now we're seeing an increase in efflux of lipids, at least particularly cholesterol. So this was really kind of interesting to think about this, and so that GM-CSF could be controlling this, and PPAR-gamma could be controlling this. And so we looked at the macrophages that lack PPAR-gamma, and we saw that they needed PPAR-gamma to allow for this efflux, this cholesterol efflux to occur. And what became really clear is when we started to look directly at the lipid droplets within these macrophages that either had or did not have PPAR-gamma, you could see a profound change in the macrophage storage of these cholesterol droplets. And so here we're labeling with the fluorescent cholesterol, and these are the macrophages coming from the tumor burdened lungs in a control animal. So the tumors are progressing. These alveolar macrophages have undergone these metabolic transformations, and you can see that they're really not storing. They're taking the cholesterol up, but it has this kind of diffuse kind of staining pattern. You can see a few droplets here and there, but now you look at the PPAR-gamma macrophages, the deficient macrophages, and you see that they're just they're handling these lipids in an entirely different manner, and they're storing these cholesterol esters, as you can see in these lipid droplets. So these PPAR-gamma deficient macrophages are unable to allow these effluxes to efflux and get transported out of the cell. And we thought this was quite interesting because perhaps this suggests an important role that these macrophages are serving to help tumor growth. Maybe this is one of the major metabolic remodeling activities that's occurring that is helping tumors to grow and progress. And that's through whether or not the macrophages is storing or effluxing these lipids. And so what we found then is that, to summarize again, that as these macrophages were becoming remodeled by the tumors that were growing within the lungs, we saw that there was an increase in GM-CSF that was signaling to PPAR-gamma that was leading to metabolic changes such as changes in lipid import, changes in efflux, promoting more efflux. And this was associated with greater tumor growth. But when we disrupted that by knocking out PPAR-gamma, or what I didn't show you by interfering with GM-CSF signaling, we saw that these changes, these lipid metabolic changes, were no longer occurring in these macrophages. They were now storing these cholesterol esters. They were also storing their triglycerides as well. And this lipid storage then active state was associated with reduced tumor growth, or tumor suppression. So we thought that this state change in the metabolic handling of lipids in the macrophages might be a very important switch that's occurring to determine how fast tumors grow and progress. So we thought this was a pretty interesting model. And this is kind of just another way to visualize this communication that we're seeing between the type 2 pneumocyte, the client epithelium that's becoming transformed in this tumor model, and its interaction with this tissue resident macrophage, where again the GM-CSF is leading to these metabolic changes. But then we are postulating that this cholesterol efflux and the lipid transport is then helping to fuel somehow the growth and progression of the tumor, and perhaps even EGFR signaling. So that kind of became our next question that we wanted to look at more was is this cholesterol efflux actually really, how is it contributing to tumor growth if this is an important role? And so we started to do single cell RNA-seq data analysis on these tumors. And we were then able to look at what was happening in the macrophages, but we were also able to look at what was happening in the tumor cells, in the type 2 pneumocytes. And so one of the set of genes that Ali started to look at were genes that were known to be regulated by EGFR signaling. And when she looked at these genes, she compared 3 different groups here. So we're comparing just a wild type healthy lung. So these are just alveolar macrophages from a lung that don't have any docs, don't have any tumor. These are then compared to lungs that are tumor burden, shown in red, so the middle column. So these are the tumor burden type 2 pneumocytes. So these are ones that are expressing the type 2, sorry, the EGFR, the oncogenic EGFR. And then we have the third column, which is now the PPAR-gamma knockout, where the macrophages lack PPAR-gamma. The tumors still express the oncogenic form of EGFR, but they are growing slower in these when the macrophages lack PPAR-gamma, and that's shown here. And so what you can see in this third column is that many of these genes involved in EGFR or downstream of EGFR signaling were actually reduced in expression. So this kind of gave us a hint that maybe there's something happening to EGFR activity, even though the model is an expression of an oncogenic form of EGFR. Maybe there's something about the EGFR activity that's being suppressed that's causing this slower tumor growth and progression. And so we looked at this a little bit more carefully by looking and staining the tumor burden lung tissue with antibodies to phospho-EGFR. So now we're looking at the active state of EGFR, this phosphorylated form of EGFR. And here we're using surfactant protein C to, again, identify the type 2 pneumocytes that have increased in their mass in these tumor models. And then this is the PPAR-gamma, where the PPAR-gamma is deficient in the macrophage and the tumors grow slower. And so here you can see there's even less tumor mass because there's fewer surfactant protein C expressing cells. But when you looked at phospho-EGFR, you can see again that there was a very broad and expression of phospho-EGFR in these tumored and these transformed cells, but you can see that the overall intensity of EGFR is much lower. There's some cells that have higher levels, more concentrated levels, but it's lower in overall intensity, and that's just shown here when you look at and quantify this data shown here on the graph on the right, that the overall levels of phospho-EGFR are considerably lower in these type 2 or surfactant protein C expressing cells in the lungs of these animals that either have control of alveolar macrophages or the ones that lack PPAR-gamma. So this did tell us that there's something happening to EGFR receptor signaling in these lungs of these animals that lack PPAR-gamma in their alveolar macrophages. So we then started to look more at phospho-EGFR in AT2 cells directly. So we were able to get a cell line. Oh, no, sorry, this we sorted out the AT2 cells. We sorted out the type 2 pneumocytes that were surfactant C positive from these lungs. And then we did a Western blot and looked for phospho-EGFR. And again, comparing the control tumor burden lungs to the ones that lack PPAR-gamma in the macrophages, we could see that there was a significant reduction in EGFR phosphorylation based on by Western blot, and that's just quantified here below. So this told us together that there was a reduction in general in phospho-EGFR activity in these lungs when the macrophages were lacking PPAR-gamma and were unable to undergo both the proliferation and these metabolic and functional alterations that PPAR-gamma drives. So we were thinking more about then the role that again, coming back to this cholesterol efflux phenotype that we saw, where the macrophages in the tumor burden lungs had been encouraged to efflux more lipids, probably again through the increase in GM-CSF signaling that's occurring. But we wondered then, what does that mean for the tumor cells if there's more cholesterol efflux going on by the tissue resident macrophage, by the alveolar macrophage? What could that be? What could that mean for a type 2 pneumocyte? So we started to dig in a little bit more into the cholesterol regulation of cholesterol within the lung airway, and came across this really interesting paper. It's like 30 or 40 years old, but they injected radio-labeled cholesterol into the lungs airways of rats, and then they looked at its half-life in these lungs, and just by collecting the bowel fluid. And what was really surprising to us is that it doesn't just disappear. Like, you would think you would inject cholesterol into the airways and it would just get into the bloodstream and just kind of go back to the liver like normal reverse cholesterol transport. But that's not what you saw. The labeled cholesterol stayed around for many days and even weeks, because it only dropped by about 10% each day. So that's how much kind of attrition of cholesterol there was. So what that told us is that cholesterol really has its own kind of almost unique life cycle occurring in the lung airways. It must be being recycled, because you can inject it and it's staying, and so these cells probably are recycling cholesterol between themselves to maintain this this labeled cholesterol that was injected into the lung airways. And we know it's going to be taken up by cells, so it must be somehow being maintained and or recycled. And we also knew that cholesterol plays a really important part of surfactant itself. Surfactant is a lipid rich molecule, but it is very highly concentrated with cholesterol as well. And I think it's the second or third most cholesterol rich lipid structure in our bodies, with the eye of our lens being the highest. And so cholesterol, again, is playing an important role in how surfactant then is being structurally regulated at the air-liquid interface. And these type 2 pneumocytes that are producing all this surfactant then have to have a supply of cholesterol to use it to add into the surfactant molecules that they're producing. And so another study had shown that about 90% of the cholesterol that is found in type 2 pneumocytes is actually coming from external sources. So they don't really synthesize their own cholesterol. They need it for the surfactant. They need large quantities of it to help continuously make the surfactant and release it to maintain our surfactant layer. But they actually don't synthesize it themselves. And I think that makes a lot of sense, because this is de novo lipogenesis that these type 2 pneumocytes are heavily involved in. And if they need to make a lot of phospholipids for the surfactant. And so we think that what they've done is they've made a trade-off, and they said, well, we're going to get cholesterol from other sources to then allow for us to focus more of our energies and our lipid synthesis on phospholipids. That's kind of just the hand-waving idea that we have as to why they might import over 90% of the cholesterol that these cells have. So they're highly dependent on external sources of cholesterol. That's the main thing, these type 2 pneumocytes. And so we then, again, coming back to the RNA-seq data, the single-cell RNA-seq data, focusing on the tumor cells, these type 2 pneumocytes, again, we asked, well, what happens with cholesterol metabolism in these cells? And we saw that there was actually an increase in the gene expression of cholesterol synthesis enzymes in the type 2 pneumocytes in the animals that lacked PPAR-gamma in their macrophages. So I know that was a mouthful. So what I'm trying to say is that when these alveolar macrophages couldn't secrete or efflux cholesterol very well because they lacked PPAR-gamma, what we saw was a compensation in the gene expression of cholesterol synthesis enzymes in the tumor cells. So the tumor cells noticed that these macrophages were no longer able to do certain metabolic functions like release cholesterol, and they compensated for that. And we think this is a sign that they're not getting enough cholesterol when the macrophages lack PPAR-gamma. And so they compensated for that by increasing cholesterol synthesis themselves. And so we actually measured this again with Christian Metallo, and we saw that indeed sterol synthesis, we could see incorporation of sterols being synthesized in these tumor cells now when the macrophages lacked PPAR-gamma in these tumor burdened lungs. So the type 2 pneumocytes were increasing and augmenting the ability of themselves to intrinsically produce cholesterol. And we believe that this is because the macrophage is deficient in providing them this cholesterol to aid in tumor progression. So we also took another approach and used spheroids of EGFR from EGFR patients, where we and these were spheroid cell lines that Sandra Liebel had made, and they were from EGFR mutant tumors. So we're using a different form, but also a commonly mutated form of EGFR, looking at these spheroids that she had developed. And we asked, well, what happens since they're dependent on EGFR activity? What happens if we now just culture these spheroids, these human spheroids, with statins for a couple of days to inhibit their own ability to make cholesterol? And what we found was that these type 2, these tumor spheroids, again, coming from type 2 pneumocytes, they were dependent over, if you chronically treated them with statins over 3 days, we continued to see a loss of phospho-EGFR activity. So in order to sustain continuous EGFR activity, you need to have cholesterol present in these cells. So I think this is another supportive evidence that these tumor cells are dependent on a supply of cholesterol, an exogenous supply of cholesterol that we would argue is coming from the alveolar macrophages. So then to just kind of summarize the major sets of kind of model that we think we've uncovered here is that we believe that there's an important relationship that's occurring between the client epithelium and in this case the type 2 pneumocyte and the tissue resident macrophages, the alveolar macrophages, and that this evolutionary relationship has developed that involves at least at one important signal, GM-CSF, that coordinates not only the development of these tissue resident macrophages but also their metabolic properties. And that the tumor cells, as they start to grow and increase in number and also increase in the GM-CSF production then that's occurring in the lungs, we see that this then alters these macrophages in a reciprocal way, where these cells now start to amplify their numbers to probably support the increase in the numbers of type 2 pneumocyte cells that exist in these lungs, but also this GM-CSF signaling is now heightening and altering and augmenting probably what is a normal homeostatic relationship and signaling process that occurs between the type 2 pneumocyte and the alveolar macrophages, but now by being heightened and augmented or even more than what's normally observed, we find that this can turn these macrophages into a tumor supportive role. And we believe that it's in part through this lipid metabolism that's occurring and the cholesterol efflux that's occurring that then allows and is able to feed back for reasons and mechanisms we don't fully understand, but somehow able to feed back to sustain the activity of the actual oncogene that then in turn is driving the tumor progression. And if we intervene in this in multiple ways, either through GM-CSF signaling, through PPAR-gamma itself, or through the alterations of cholesterol production through statins, that this can then lead to a suppression of tumor growth. And with that, I just want to come back to this model that again, coming back to this model of the kind of founding concept that we believe that there's these evolutionary homeostatic relationships that can occur between the client epithelium and the tissue-resident macrophages that might be getting co-opted to allow for these macrophages to have greater assistance and support for their client epithelium. But in this particular case, in the case of cancer, that these functional changes that are being induced by the client epithelium and signaling to the tissue-resident macrophages can turn the tumor, the macrophage, into more of a tumor promoting state to allow these client epithelium that are now malignant and transformed to continue to progress. And so with that, I'll just end, and thanks Allie again very much for all of her beautiful work and our collaboration with Katie over the years. And of course, Christian and other people at Salk have also been very helpful to this. And this work has been funded by NIH and the Mark Foundation and Cancer Research Institute and the NOMIS Foundation. So thank you very much.
Thank you so much for a great talk. I would like to ask a couple of questions first, and then let Jennifer will field the rest of the questions. We've been working with a KRAS mutant lung cancer model. Have you also worked with that model? And if so, have you seen any differences in these pathways in KRAS mutant versus EGFR mutant tumors?
Yeah, so we have been doing that with Ruben Shaw. It's not, we haven't done a KRAS specifically. We did a LKB1 mutant lung tumor, and I don't think that had KRAS. He has a bunch of different models. I don't think that had KRAS in it, but it no, it must have. Yeah, it had KRAS and LKB1. Yeah, and we did not see the profound increase in the alveolar macrophages that we're seeing here. So that could be interesting to think about. Are there going to be oncogenic types of, genotype specific effects on the alveolar macrophages? We weren't able to measure GM-CSF levels in those tumors. I think that would be a really interesting thing to do. But we did see an increase in neutrophils in those tumor models, which has been reported before in LKB1 mutant tumors. So, which in humans and in mouse. So we did see some effects on the myeloids, but we didn't see this dramatic increase in the alveolar macrophages.
So I would just say that we've been working with a couple of KRAS mutant tumors, and we see a huge increase in alveolar macrophages. And it's not GM-CSF driven. So we're getting ready to submit that. So I'd love to compare notes with you.
Yeah.
They are lipid associated, but so one of the things I'm curious about is whether there is any disruption in the surfactant layer in these tumor-bearing animals, because you would think there would be, and I'm curious about.
Yeah, I think there is for sure. The total abundance of lipids are increasing, and we looked specifically at the phospholipid that's most abundant in surfactant and DPPC, and that is also higher in these lungs that are tumor burden. And the one of the features of PAP, when you see the proteinosis, you see is that the bowel fluid becomes very milky when you physically take it and look at it. And we also see that in these very late stage tumors. We haven't used that as like a time, we haven't longitudinally sampled this to see how it changes over progression. But at these end stage, when these animals will be dying if we don't sacrifice them before because they just can no longer breathe. So the tumor burden becomes so massive. So I'm sure there is a major alteration in surfactant quality as well.
Just one other quick question for the benefit of everybody. This tumor model is a Cree-driven model? No, it's a
doxycycline induced.
Right. But there is a tissue-specific promoter being used?
Yeah.
And is that SBC or is that?
No, it's in the Claricell promoter. Yeah.
Okay, great. Well, thank you very much.
Yeah.
So I might ask a quick question before we ask from the general audience. So in the PPAR-gamma deficient setting, those macrophages that have increased lipid retention, I guess, what happens to them? They just become full of lipids. And so first question is what happens to them, but then the second question is, can you also induce that state with using a PPAR-gamma inhibitor or a statin or some other drug like?
Yeah, so we didn't look at the statin treated lungs. We did treat with statins and showed that I should have added that slide in. We did treat with statins and the PPAR-gamma deficient macrophages and then compared that to the control. So if we just give statins alone, we saw a small effect on tumor progression, but it wasn't really significant. But when we then treated statins in the PPAR-gamma deficient, when the macrophages lacked PPAR-gamma, those tumors were much more sensitive. We saw hardly any tumor growth. So the combination of statins, that again told us that the tumor progression was much more highly dependent on cholesterol synthesis when the macrophages lacked PPAR-gamma. So that was kind of another indicator that this compensation that the type 2 pneumocyte was happening was dependent. But to the antagonist, we did look at all those metabolic changes that we saw, and they have the same effect. So when you give the PPAR-gamma antagonist, there is an increase in the lipid droplets in the alveolar macrophages. So
and what happens to those?
They're becoming foamy macrophages in the absence of PPAR-gamma. The long-term effects, we don't know. We've never looked at viability of those cells, but they have all the features. They're more inflammatory. I didn't show you that. There's a lot of data I didn't show you guys, but they're more inflammatory. They have more TNF alpha when they lack PPAR-gamma. So they have these kind of foamy macrophage attributes. Yeah.
Very cool. Thank you. Jelani, you had your hand up.
Hi, Sue. Well, wonderful talk. I just had a question about when PPAR-gamma is knocked out. Have you looked at those tumor cells to see if they upregulate the Kennedy pathway to making new lipids de novoli? So the Kennedy pathway is phosphatidyl choline to phosphatidyl ethanolamine. So it's a process in which cells can make new lipids de novo. And I was wondering if that's a way that those lung tumor cells are able to upregulate new lipids even when TAMs lose PPAR-gamma.
Yeah, we didn't, beyond just the RNA-seq data, we didn't look at those, the pathway that you're looking at specifically. The cholesterol pathway was the one we saw kind of show the most augmented activity. But I think that was, we would love to do, just like to ask now, was there a trade-off? Kind of getting back to Judy's first question, what happens to surfactant? Is there a trade-off? If now they're forced to make more cholesterol, do they then now have an impaired ability to make phospholipids too? Like is the amount of phospholipid synthesis going down? So I think all the, and actually I think what you're raising is a point I also meant to say at the outset, which is that I feel like cancer biologists, we've studied cancer metabolism for decades. We've learned so much about cancer metabolism, but what I don't think we think enough about is what were the metabolic activities and properties of the cell before it became transformed? So the healthy state of that cell, what are those metabolic properties that those cells have? Because those are probably going to be carried out in, even as the cell becomes malignant, certain metabolic properties are going to be, and activities are going to be manipulated and augmented to allow for, increased growth and an invasion of kind of tissue, tissue control pathways, but they're probably imprinted with, certain features of that cell state before it became malignant. And how many of those metabolic, so I think, I don't know how much we know. I don't think we know a lot about a type 2 pneumocyte metabolic state to begin with, and how it's fluctuated in different contexts, to then think about, well, now what's happening, what's different when it becomes transformed, and that's why we thought like this, thinking about this kind of evolutionary relationship between these cells and their metabolic relationships is really important, because they're probably just the ones they use every day, but now they're getting modified. And if you can intervene with it, then you'll maybe find vulnerabilities in the tumor itself because it has metabolic dependencies on these tissue resident macrophages.
Thank you.
Yeah.
Shelley, Arp? We had you for a second.
Hi, Sue. I just got back from the 40th anniversary of the cloning of the EGF receptor. So the most startling thing is the loss of EGF receptor phosphorylation. The mutation didn't go away.
Yeah.
They've got an explanation for that. One of the potential is that you're increasing the phosphatase activity as a result of the lipid changes. Do you have any other ideas?
No, we don't. I think that's really interesting. And you're right. The protein level is there, right? Where, I mean, here is a DOX-inducible gene expression system, so it really was the phosphorylation. The only thing we can think of is, our simple mind was that it has to be something with plasma membrane structural aspects of how the EGFR receptor is dimerizing or something like that. And when we dug into this, it was a little bit confusing at first because when people have looked at what happens when you take cells and you deplete cells of cholesterol at EGFR signaling, but almost all of these studies that we could find had looked at very short-term treatments, like after 10, 15 minutes of cholesterol depletion, you actually initially see an increase in EGFR signaling by just eliminating cholesterol from the plasma membrane. And that is probably has to do with the dimerization aspects of it. So that is initially what happens, but then what we're looking at is sustained depletion over many days. And then that's when we started to see it drop. And of course, those spheroids, those cells actually will start to die because they need that EGFR signal to stay alive. So that we had to, we couldn't deplete for too long. We lost the cell, or we couldn't treat with statins for too long, or the cells they just all died. But we think that means that you need the presence of cholesterol in the cell. Another could be internalization. We didn't really look, well, I mean, these are ligand independent, so I guess that wouldn't really make too much of a sense. But yeah, we kind of scratched our heads. We don't have a good mechanism to why cholesterol's helping to maintain the phosphorylation.
Well, phosphatases are evil, so look
Yeah.
there.
Yeah, that's a good idea. Yeah. Thank you.
We have some questions in the chat. There's a question about the phagocytosis of neoplastic cells by the alveolar macrophages and the effect of the metabolic phenotype, and then how PPAR-gamma knockout would they have decreased phagocytosis?
Yeah, we looked at that. We looked at that a long time ago. I don't remember. I think they did have a decreased phagocytosis. I don't remember. Sorry, it's been a long time since I've looked at that data, but yeah, my memory is that they did have decreased phagocytosis. I remember when we first started on this project, that was like one of the things we looked at, and I can't remember. I haven't looked at it in a long time, but I think that's what we saw. Yeah.
There's a question about if you've checked the significant differences in metabolite contents, including lipids in the extracellular matrix. And they're specifically asking whether cell-free metabolites and lipids are circulating differently between healthy lungs, early and late-stage tumors.
Yeah, and by extracellular matrix, do you mean true extracellular matrix?
Probably just outside the cell, right?
Yeah. So sorry, can you repeat the question again?
Yeah, are there differences in cell-free metabolites and lipids that are circulating differently between healthy lung, early and late-stage tumor progression?
Yeah, we focused on lipid metabolism. We didn't do a total metabolite profiling, like just kind of steady state metabolite profiling. So I can't say what other metabolites might be different, but the lipid profiles were what we looked at the most, that were increased in many different lipid classes. Yeah.
And there's questions by Samantha Hedge and Fulvia Bascotto. Do you want to turn your cameras on and ask those questions? Otherwise, I can ask the last two. Okay, so Samarth asks, how does the accumulation of the TA tumor-associated alveolar macrophages reconcile with the exclusion of resident tissue macrophages observed in lung cancers that are shown by the Murad or Amit groups, with the paracrine effect of cholesterol exchange play a role?
Yeah, we definitely have thought about that. We just don't, so her model's a bit different. We just don't see the infiltration of a lot of bone marrow derived macrophages. We don't see many interstitial macrophages in this. So I don't think this has anything to negate what she has found. I think what both studies show is that these tissue resident macrophages can be operating in tumor promoting states. In her model, they were physically excluded, and then the bone marrow derived macrophages were surrounding the tumor mass more. We just didn't see that in this model. I don't know if it's because of where the origins of the tumor emerges from, in this particular case, strictly from the epithelium versus the models that she used were lentiviral models that can, a variety of cells can become the origins of those tumors, but that could be one difference.
I'd like to say we like you, we see the same thing. It's almost all driven by alveolar macrophages. Very few inflammatory macrophages, even through the late stages. So there's a difference in
and we did
the human. I think when you implant tumors, you induce an inflammatory reaction.
Yeah,
I think that could be the difference.
Yeah, and we also we profiled some of the as our work was progressing, more and more single cell RNA-seq data came out, and we did find other studies that had found that some studies had found an increase in alveolar macrophage density based on the single cell RNA-seq, and some didn't. But there was an increase in PPAR-gamma signal. And that was pretty consistent across the other human lung cancer samples. So,
could I just ask one quick question? I don't know if you've done much pathology, but can you tell a difference morphologically between alveolar macrophages and others? I mean, we think we can, and I just want to make sure it's not because of where they're located, but it's actually the cells themselves have a different.
Yeah, well, I can tell you, we can see I guess we never asked the pathologist to distinguish between alveolar macrophages and other macrophages, but we definitely see them along the airways, but they are inside the lung parenchyma in these tumor burden stages too. They're not just in the airways. So they're moving inside and interacting with other cells that they probably wouldn't normally be interacting with. I don't know if I don't think we ever stained. We focused we did do a ton of histology. We can definitely give you the slides to look at if you have a better eye for that, because what we focused more on was the actual tumor cells and trying to ask, because when we knocked out PPAR-gamma and the macrophages, the tumors, I mean, even the morphology of the tumor cells was different, which could be an indication, of some of the things that are going awry when they don't have that support from the macrophage. But
so we've done a lot of lung work lately, starting with our work on COVID and now on these lung models. I would say that lung alveolar macrophages are really different, and they're huge, they're round, they have a really different morphology. And when you see inflammatory macrophages, they're more they look more like fibroblasts. They're like squished between cells. And so it's really striking. And even when there's a lot of macrophages that are surrounded by tumor cells, to us, they seem to maintain the same morphology, which
I would agree with that. Then, we still see these kind of round, fluffy cells. Yeah.
And I wonder whether their lipid metabolism properties don't have something to do with that. So I think it's something to consider. Morphology significant.
Yeah, it would be interesting to look at our PPAR-gamma knockouts then and see, because we definitely can see the lipid droplets, and they look like foam even under a bright field. We can see that they have a foamy kind of appearance. So I don't remember if we looked at that in I think that came out of the histology too, but I'd have to go back and look at that.
We have one final question from Fulvia.
Hi, can you hear me?
Yes, we can.
Yeah, Thank you. I modify a bit the question, let's say, because I'm wondering if you have tried sorry if I missed any treatment of the mice bearing the lungs with the metabolite to convert or to skew the effect of the tumor-associated macrophages.
Yeah. Well, we treat with the PPAR-gamma antagonist, and I meant to say that there's a company that's making PPAR-gamma antagonists now, and they're going into some clinical trials. So I think this is interesting. Most people would never think about a PPAR-gamma antagonist because they actually were, years ago, these were the agonists or what we use for type 2 diabetes. So an antagonist, but kind of our rationale was like, well, maybe if you could deliver it very controlled and locally to the lungs, it might not have systemic effects, and it could be an interesting way to think about manipulating the macrophages. It might even in the simple role that it plays in just alveolar macrophage development, if just reducing the overall numbers of mature macrophages, as our genetic models suggest, that in itself could reduce tumor growth. So we think that could be an interesting thing to think about. And we were also very interested, and we haven't been able to get a clinician to look at this with us, but coming back to this connection with statins and TKIs, right? So if EGFR receptor is inhibited by TKIs, if we gave statins, would that have an augment effect? And statins, being used to treat cancer has been in favor for many years because the role that then the whole melatonate in cholesterol synthesis pathway has in creating farnesylations and all these things that will help to maintain RAS signaling. So there's all these other signaling roles that this pathway can play, and so I think combinations of statins with TKIs would be a really interesting connection to look at, maybe with PPAR-gamma antagonists, so.
And Shorty, just one comment also on the observation that you see the proliferation of macrophages, alveolar. How much really is proliferation on those cells? Do you know more to describe how really much they proliferate?
Yeah, only that we measured proliferation by Ki-67, and that was higher in the tumor burden lungs. So they seem to be in an active state of proliferation. And when we treated with PPAR-gamma antagonists, that population of Ki-67 cells did decrease some. So that's all we really have to.
Absolutely. Thank you.
Yeah.
All right. Well, thank you so much, Sue, for a fantastic talking and a great discussion.
Thank you. And thanks for organizing this also and all the time. I know from Global Immunotalks, this is a very time intensive process. So thank you so much. Yeah.
Thank you.
All right. Bye. And Judy, I'd love to, let's get together and talk more.
I would love to.
Yeah.
Okay, great. Thank you.
Happy holidays, everyone.
Yeah, you too. Bye-bye.
Bye.